Hello, LS students. How are you today? This is week 6 and we are going to talk about the synapse. A synapse is a gap or junction or connection between two excitable cells, a neuron and another structure, which could be a neuron, a muscle, or a gland. The neuronal pathways, it could be divergent or convergent. In a divergent pathway, one presynaptic neuron branches to affect a larger number of postsynaptic neurons, while in a convergent pathway, many presynaptic neurons converge to influence a smaller number of postsynaptic neurons. By observing a neuron-neuronic synapse, we can see that we have axosomatic, or the junction between an axon of a first neuron and a cell body of the second. We can observe also axodendritic, an axon of the first with a dendrite of the second. Axonic, it's between two different axons. The synapse consists of the presynaptic neuron, the synaptic cleft, and the postsynaptic neuron, which could be a muscle or a gland. Structure of a synapse. Here we have the presynaptic membrane containing the synaptic vesicles. In between we have the synaptic cleft. Here we observe exocytosis, or the release of neurotransmitters from the presynaptic membrane into the synaptic cleft, then into the postsynaptic membrane. Steps of synaptic transmission of a nervous message. First, we have arrival of action potential to the terminal buds. When the nervous influx reaches the terminal bud, CA2 plus channels open and comes in causing depolarization of the membrane. Next, there is exocytosis, which is the release of that neurotransmitter into the synaptic cleft. These neurotransmitters will bind to the receptors of the postsynaptic membrane. The binding of the neurotransmitters will cause the opening of ionic channels. Two types of neurotransmitters in this case. We have excitatory neurotransmitter. In this case, it will cause depolarization of the postsynaptic membrane and leading to the formation of an action potential if membrane polarity reaches the threshold potential. And we have the inhibitory neurotransmitter, which causes hyperpolarization of the membrane, and in this case there is no action potential. The binding between neurotransmitters and receptors may have two effects, depending on the neurotransmitter. If it opens the sodium channels, Na+ enters in causing depolarization of the postsynaptic membrane. An action potential is formed, a nervous influx, as you see in this figure. While if it opens Cl- channels, Cl- ions enter inside the neuron. Hyperpolarization inside the neuron becomes more negative, or K+ channels moves out. In this case, hyperpolarization of the postsynaptic membrane, the reading would be -100 millivolt, and no action potential would be recorded, and therefore no nervous influx. As you see in this figure, we have a reading of -100 millivolt. If the stimulus opens K+ channels, this will lead to graded potential or hyperpolarization. However, if the stimulus opens Na+ channels, we have graded potential called depolarization. And if it leads to stronger depolarization stimulus, it will open more Na+ channels. In this case, an action potential is recorded. Then the neurotransmitter is either degraded by the enzymes of the synaptic cleft or recaptured by the presynaptic membrane in the terminal bud, or can diffuse out of the synaptic cleft, as you see in this figure. Finally, I hope you have a great day. Thanks for your attention.